Well, thank you very much, Dean Curtis. It's a really great honor to be invited to UC Davis and meet my friends, and then all of the ones that have emigrated from the Midwest have come here to greet me. So I feel very much at home. Thank you very much. And really enjoyed talking with the faculty yesterday about some of the exciting programs that are going on here. And this afternoon I have a real treat. I get to meet with the graduate students as well. So it's really great. So what I'd like to do today is just discuss a little bit about my research, which really addresses the fundamental area of proteins at interfaces. But it's interfaces of proteins with themselves, with solid materials, or with the human body. And so I'll try to connect this theme and show you some of the things that I've done over the course of the last few years. Let me see here. Suffice it to say, in order to do research and support students, you need funding, grants. And so I've been very, very fortunate to be supported by industry, by Purdue University, and the stakeholder groups we have. We're also, like Davis, a land-grant school, and also government agencies. So this is a picture of my group. Not all of that, but this is the core group. I might say that 4 of them have graduated and moved on already, so the picture would be different this year. But the key people that really enable me to do the work that I enjoy are the students. And so I'm very grateful, and I thought a picture would be helpful. There's many that are not listed, and they're on the right-hand side here. So let's start out by examples of biofilm formation. And this has to do with produce, and the fact that with produce and our move to eat healthier, we also have incidences of pathogen and people getting sick. And so the question is, how does this occur? How do you detect it? And then eventually, how do you minimize or prevent it? Well, it turns out plant tissue that's been macerated by naturally occurring enzymes, proteins, also provides a way of pathogenic microorganisms, particularly salmonella, to grow more quickly and basically to outcompete natural microorganisms. E. coli, the pathogenic type, and these all exist on produce, on cantaloupe, as you'll see in a minute. And by the way, if you go to the store and you wash it carefully and they have the produce wash, you're fine, okay? It's only when you don't wash it that there's a problem there, okay? And if it doesn't look right, then return it. Okay, so if you have a film on it, you'll notice. But anyhow, sometimes what will happen is these films will form, and E. coli, pathogenic type, 0175787, for example, will in fact be sequestered in these films and protected against being washed off, and also very difficult to detect. Salmonella, the same thing. And so you have produce or leaves, and they form a film. Well, biofilms are not new. They're formed on biomedical devices. These are pictures, Brian might relate to this. These are pictures of biofilms forming on mild steel. So when you manufacture pharmaceuticals, for example, you want to make sure there is absolutely nothing in the final makeup water. It's called WFI, water for injection. So very much concerned about microorganisms forming on mild steel, and in fact they do. This is a picture after 8 weeks. On your right is a picture of microorganisms on stainless steel, and these are dyed. And again, the blue color are the microorganisms. And the reason they're able to do this and survive is they don't need a lot of nutrients, and once they grow, they form exopolysaccharides, like in this picture, where for produce you have something that's even a little bit easier. You have organic matter so that they can, in fact, produce these exopolysaccharides. And this is a E. coli biofilm on lettuce. And then on your right, this is salmonella on cantaloupe. And the reason there was an outbreak of salmonella possibly is when you cut the cantaloupe before washing it, you swipe that outer surface of microorganisms into the fruit. And so again, there are procedures that can be used, and I'm sure California and the FDA, FDA is the agency that regulates produce, are working very hard at it. So the outbreaks are very, very rare, by the way, but once they hit, it is a big problem, and you'll read about it. So what can we do about this, since we know the films form? And so the way the film formation occurs, you get contamination from groundwater, from improper agricultural practices, from organic farming if the manure is not sterilized. These are all possible sources. Most of the microorganisms that are there are not pathogenic, but if they are there, they can get on the surface, they can proliferate, aggregate, and then they form a biofilm that protects them from other competitors. And finally, you get bacterial dispersion, such as when you harvest or you would consume it. And this is called, a big word, microbial translation to humans by plant vectors. So what this says is plants can in fact be vectors of human disease if a film forms. So how do you break these down? It requires enzymes. And it's a little bit interesting. For pathogen detection, I'll get that part of the story in a minute, we use proteins, enzymes, to help detect these pathogens. But the pathogens themselves have to be living. We can do PCR. You don't need a living microorganism for that. Okay? And so as you collect or capture these microorganisms, you also have to make sure they're alive. Second of all, the pathogens are the minority of the microorganisms. Most of them are not pathogenic, many are helpful. And so you have to be able to fish out these pathogenic microorganisms from the rest of the population. So the recovery of living microorganism, you can grind it, you can use enzymes to break up the biofilm, you can use enzymes to break up the cellulosic structure of the produce, and where I spent part of my career is cellulose hydrolysis for making biofuels. Guess what enzymes we use? They're the same families of enzymes, except for a different purpose. The other thing that's very interesting is there's extracellular DNA, which is quite prevalent, which is also part of the biofilm. And so you need DNA, restriction enzymes basically, to break up the part of the exopolysaccharide that is DNA, again, to release the microorganisms. So recognizing that, the next step is, in order to be practical for detecting pathogens in foods, you have to be able to do it relatively rapidly. Right now, standard techniques which involve culture require up to 3 days. What we want to do here is be able to detect the pathogen, if it's present, in about 1 work shift, less than 8 hours. Okay? So the question is, how do you do this? And so the culture step these days is really the time-limiting factor. The culture amplifies a pathogen to the point where it can be probed or detected. So what we've done instead is do microfiltration, where we can do the same thing in about an hour. And so that helps reduce the time required in order to detect the presence of a pathogen, should it be there in the first place. So releasing, concentration, and recovery involves several steps. First of all, you get the sample. The FDA lab, for example, if they're doing cantaloupe, will have buckets of water in which they plop the cantaloupes. They wash them off. Then, because they have so many samples, they will combine samples from each of these and test them. And they'll have, let's say, 100 buckets in a day. And if they find any indication of a pathogen in one of the triage samples, they'll go back and check every one. So there's some statistical and other methods used in order to do detection in an efficient way. Then after that, we pre-treat. So we incubate this material with enzymes so that the microorganisms are released. And then we use a different set of enzymes to prevent microfiltration, the microfiltration membranes from fouling. So there's 2 different steps. We then pre-filter to take out the big chunks. And then finally, we do the cell concentration recovery, where we take 225 milliliters, concentrate it down to several milliliters, stick it in a microfuge for 10 minutes. We get a nice pellet. The pellet includes solid particles that are not bacteria, but also has a very high concentration of all bacteria, most of which, by the way, are not pathogenic, and most samples of which do not contain pathogenic cells. And so you have to have a high enough concentration to efficiently interrogate them and get it done in less than 8 hours. So what we proposed to do, and this is a few years ago, I think you may have still been at Purdue, I don't know. Or maybe you were at University of Florida. You have a very famous dean, by the way. And I see the department head from Biomedical Engineering here as well. And then, of course, Brian Jenkins. So I'm really just so... Where's chemical engineering? Oh, there you are. Okay. So I'm really flattered that you would come. So anyhow, so what we want to do is take the shaker flask and the Petri dish approach, which is standard microbiology, works very, very well, by the way, but takes 3 days, and replace it with hollow fiber microfiltration, where we extract the sample, we extract the bacteria, we have to be careful how we do that, and then concentrate the liquid from a large volume to a very small volume, and then we use modern techniques that only require small volumes to probe the sample to find out if a pathogen is present. And we can use PCR if we know it's living, or we can use other methods, spectroscopy, in some cases mass spectrometry, okay, but we need a concentrated sample. So these hollow fibers are between 200 and 300 microns in diameter. You can see my finger. And so the other thing we have to do is be able to pre-filter the big chunks out before we go to this step. So we started by purchasing the hollow fiber from a company in Michigan, and then making small tubes that are about the size of a pen, which had between 3 and 20 or so hollow fibers. And here you can see it. And then on the inside surface, there would be a coating. And the way the cutoff of the hollow fiber, which was between 0.2 and 0.45 microns or so, was due to depth. So if the cutoff is a lower molecular weight, the depth is higher, right? Because it's more tortuous. So these are not through pore, but rather depth filters, fabricated in a very small diameter of the hair, roughly, fibers to give you a lot of surface area per unit volume. And then the way it works is you have a feed pump. Oh, here we go. It goes through something that looks like a straw. It's recycled, goes back into the feed concentrate reservoir. Of course, we have media to help the microorganisms survive and grow while we're doing this. We've added enzyme to prevent fouling. We've also very carefully selected the enzymes so the enzymes don't kill the pathogen, which is what they tend to do, so we can detect them if they're there. Remember, a dead pathogen does us no good if we're trying to detect this for purposes of food safety. So after about, depending what it is, between 10 minutes and an hour, the recycled fluid is reduced in volume. The cells are hopefully still there. We then collect it as a concentrate, and as long as they're viable, we can then go to the next step. So microfiltration, you might say, as us chemical engineers know and ag engineers know, is not new. It's been around for 70 years, but fouling has been a huge issue. So what we decided to do is use enzymes in order to help us prevent fouling. And the way the fouling occurs is a small molecule, it's 0.2 to 0.45 microns, so a really, very large cutoff. And the molecules that we're trying to separate the cells from are probably on the order of 100 kilodaltons or less, so they're very small. So you'd think they would go right through the pores. They don't. They get into the pores, and then they start to interact with one another. And because the microenvironment is different, they will basically precipitate or aggregate and plug up the membrane. And if you're working with milk, for example, it takes about 1 minute. The dead volume of these hollow fibers is very small, so if you pass 1 mil through, it's probably, 20 or 30 dead volumes. That's enough to plug them up. And so what you have to do is find some way to prevent that. So we used addition of enzyme. This was Dr. Eduardo Shamanis' idea. He's very good with enzymology and molecular biology of large proteins. And so as a consequence, we went to the literature, and lo and behold, there's all sorts of enzymes that have been used for similar purpose on a large scale. And you can read the enzymes. We also added to this list cellulases and lipases. Okay. And what they've done is use this for microfiltration. That's what MF stands for, or ultrafiltration, which is basically a molecular weight, probably, depends on the size, less than 200 kilodalton. Enzymes have been added to prevent the fouling and the blockage of the membrane. So that's not new either. What's new is the combination of the media, getting the cells to grow, selecting the enzyme, selecting the conditions, avoiding fouling, okay, and preventing the enzymes from killing the cells we're trying to detect. So by combining all that with another method, and that is, this is more on hydrodynamics, That is to keep the pressure drop as the microfiltration is occurring. You have a long fiber, right, and the fluid is going down. Well, as the fluid goes down, some of it goes through the wall. So that means the control volume of the fluid is decreasing. It's unsteady state as it passes through the hollow fiber. So that means the pressure drop as you go from one end to the other is dropping. So at the end, there's a lower pressure drop. Well, things slow down. Okay, you don't have enough surface flow, so they start to block the membrane, and then it just kind of goes backwards, and before you know it, you're not able to filter. So what someone else figured out is that by keeping the pressure drop constant as you pass the fluid through that hollow fiber, you're able to minimize this other cause of fouling. So the way we did it is we have flow on both the inside and the outside of the hollow fiber. So next step was we put it together. We've built 6 different versions of this particular instrument to show that all of these things could be put together. And so this is controlled off a lab view. All right. And what you can see is it's an instrument that does 2 samples at a time. And it has 2 hollow fibers. This is a hollow fiber. It's got 2 pumps. One pumps on the outside, one circulates the fluid on the inside. Okay. And the first step is prefiltration. So this is your 3rd pump. By the way, Kirk Foster built all this. I didn't. I just traveled around. Alyssa knows who Kirk is. So Kirk Foster is probably one of the best instrument makers in biomedical engineering that I know of. But he was superb. He built this. And so here's an example. So we start, this is for spinach. We start with the fluid at 60 mils. This is a smaller sample size because we inoculated with a larger amount of microorganisms initially. And here's the waste reservoir. So after 4 minutes, we've microfiltered about 45, 50 mils. And you'll notice that they don't all filter at the same rate because the samples are a little bit different, the hollow fiber is a little bit different, and so there has to be some control so that you stop at the same point. Okay? But still in 4 minutes, and you can see the fluid that's been collected. In 6 minutes, we're collecting the sample off the tube. Then we take the tube off. The reason this person has a mask is Salmonella and E. coli, and some of these require a BL2 safety hood. Because the instrument was so large, it would interrupt the airflow in the safety hood we had at the time. We had to make the whole room BL2 compliant. And that requires use of mask, physicals for air function, for lung function, all sorts of things. Okay, but so we followed the safety procedures. He decanted a microfuge tube. He put it into a microfuge, spun it down, and then what we did is we ran a number of tests. So what we've done so far is from 25 grams of spinach, which is a standard protocol, where we spike Salmonella into it at equivalent of 1 CFU per gram, which is very low. And then we extracted the spinach into 225 mils of media, which is a standard volume used in many of the pathogen assays. We then concentrated, collected the sample, and ran PCR. And the PCR showed for this 1 cell per gram that Salmonella was present. This is a positive control. This is a negative control. This is your standard showing the molecular weights. Okay? So what this shows is DNA that's associated with the Salmonella was present in that sample, where the sample contained millions and millions of cells and 1 Salmonella per CFU. Some of the standards for the government is detection of 1, not just 1 per gram, but 1 microorganism, which is a really tough standard, which is why growth and microbiological techniques are often used. The other thing we found out is we do have to have a growth phase. We do have to have media in the instrument to grow the cells, because if the cells are stressed, their phenotype will change. They will no longer express the proteins that identify them as being pathogens. And so you have to spend some time in order to grow the microorganisms to basically resuscitate them so that their true character comes out. Okay? And so that's the other reason we do that. So this is all done while we're processing the sample, because by the time you sample it, you extract it, you filter it, you put it through the C3D, there's 2 or 3 hours. And so we pick a media where the cells that have a doubling time of 20 to 30 minutes will continually grow as we're processing them. The media we use is selected for the pathogen. Okay, so that's part of the overall system. It's all packaged into a nice buffer, nice set of enzymes you sprinkled in, and away you go. So when we went to FDA, and we're working with FDA on this, and they have a tremendous group, and they've been very kind to us. They're very busy people, but they gave us the access to talk to their research scientists. And rather than have us tell them what we think they should do, we tried to listen to find out what they really wanted to have in their lab. And one of the things they want is something that's multiplexed. So the next step is to build a machine. We started with 1, 2, now to build one with 4. And this is what it looks like. Okay, so now we can process 4 samples at a time. We're now moving on to 8. Once we have 8, we can stack them, we have 96. You know where this is headed, right? And so the issue we now face is the volume of fluid we have to handle because of the protocols. 225 mils is your first sample, becomes huge. And so we're still working on this. But we're working with FDA and with industry to streamline some of the processes. So it's now a microfiltration issue. And again, it's because we were able to learn from the literature, from ourselves, and from our research to minimize fouling. So that then takes us to another part of a very interesting area, and that is lignocellulosic conversion. Again, using very similar enzymes. So in this case, if you look at lignocellulosic corn residue, wheat straw, switchgrass, which is popular here in this area, wood chips from wood that is not suitable for lumber or for pulping, etc. They all have about the same composition. They have glucan, forms glucose, silane, forms 5-carbon sugars, lignin, and some other components. And it turns out that you can convert these sugars into ethanol, and it gives you roughly about 100 tons, in best case, 100% efficiency, 100 gallons, 400 liters, roughly 380 liters of ethanol per ton of wood or corn stover or wheat straw on a dry basis. So this is a very good source of fermentable sugars, but to get at it, you have to look at the cell wall structure. The cell wall structure consists of lignin, cellulose, which is crystalline, hemicellulose, which is again a polysaccharide, but it has a branch structure. So schematically, what we're showing here is you have lignin that's protecting the cellulose from hydrolysis, the hemicellulose, which gives it tensile strength. Okay, and by doing pretreatment, what you do is you open up the structure, you melt the lignin a little bit, you dissolve, partially break down the hemicellulose, and then open the structure of the cellulose so that the enzymes can access this. The catalyst can access it to break down the cellulose polymer. It's a linear polymer of glucose into monosaccharides. So that's what pretreatment does. So the pretreatment we've worked on is liquid hot water. The very first pretreatment I worked on with Chris, actually, interesting, was in 1977, was an ionic fluid called katoxin, which is not something we would even consider using, but from the point of view of principle, we mixed the katoxin with corn stover and then added cellulase enzymes at the time and got very good hydrolysis, which told us that if you could breach the lignin and decrystallize the cellulose, the enzymes could in fact give you very good conversion. Over time, we gravitated towards something that was taken from very early pulping literature over 100 years ago, where they noticed that cellulose pulp cooked in liquid hot water seemed to be more reactive. They called it alpha cellulose. So we studied that and found if we could separate the pretreatment step, opening up the structure from the hydrolysis, we could prevent the formation of aldehydes which come from the monosaccharides, prevent the formation of inhibitors of fermentation, and maximize the amount of sugar we get from the biomass material. And these are the conditions, 160 to 215 degrees C. Our first publications say, gee, you really want to keep the pH around 7. We found out very quickly because of the soil that is found in some of the biomass or the silica, it naturally self-buffers anyhow to 4 to 7. So you get a little bit of degradation. So you don't have to add any chemicals either. So then the second step is to use enzyme, which goes from a cellulose that's accessible, that's the C star, to glucose. And because the enzymes are so selective, they operate at 50 C, there is very little degradation product. So the reaction is selective, more than 99%. If you do the pretreatment correct, you also get a high yield. The idea here is we don't want to add any chemicals we don't have to. First of all, it sounds nice, it's natural, it's green, but actually it costs money. Because when you add the chemicals in, you got to pay for them, and then you have to remove them at some point, and you have to pay for them. And that's something I learned separately when I tried to scale up some of this technology. So if you do pretreatment, this is from the very early days. For corn stover, for example, the red line on the bottom, no pretreatment, very little conversion. The blue line above, pretreatment. The conversion, I think, at this particular experiment was 70%. We combined enzymes from DuPont, Danisco, and Novozyme. Have anybody from Novozyme here? Oh, I'm on tape anyhow, right? So hello. So I know Novozyme has a research center here, right? Really tremendous research center. So yeah, the blue line is hydrolysis, but we combined enzyme from DuPont, Danisco, and Novozyme at the time. It was 15 FPU or higher. So the total enzyme loading was more than 25 milligrams per gram. Economic, you're trying to reach 1 or 2 milligrams per gram. So this was not economic, it was to prove a point. But turns out when we combine the two enzymes, which seemed to work very well, we were combining a beta-glucosidase, Novozyme 188, with a cellulase from T. reesii in a combination that in fact resisted inhibition by phenolics, we now know, that were thrown off by the lignin when the lignin was in fact pretreated. Oh, so you've been there, right? You're shaking your head, I guess. Yeah. Oh, okay, so just following. I thought maybe I had someone else who spent their life doing lignin. You know, do other things too, right? So in an overall process context, you have pretreatment, then hydrolysis, fermentation. If the hydrolysis and fermentation is combined, as Lee Linnet had proposed, it's called consolidated bioprocessing. And there are now yeasts that, in fact, produce cellulase enzyme and also ferment the sugars that are formed to ethanol at the same time. You then have distillation. The solids which remain, which are lignin, right now are then used for generating power. And because of the energy content of the lignin and the efficiency of processes, you actually get more energy from the lignin than you utilize in the process. So they have combined heat and power, CHP. The issue then is most of these plants have to be built near the biomass source, and the biomass source usually is not near an electrical grid. So then somebody has to pay to bring in the electrical grid, which is very expensive, to remove the electricity that would be generated by burning the lignin that's left over when you have all the requirements for the plant. But the way pretreatment was done in the lab is in a tube, 37 mils. You've seen these before. You don't fill them up all the way because if you heat up water in a liquid state at the saturation vapor pressure, it expands, right? If you fill it up all the way, well, you won't do it the second time. It's a lot of pressure, right? A lot of hydraulic pressure. So you fill them up about 2/3 of the way. You put the biomass in. We do anywhere between 100 grams per liter to 300 grams per liter, depending what it is. You cook it in a sand bath. To quench the reaction, you drop it into a bucket of water, room temperature, and then you collect the material and do an analysis. So that then, you remember this, Alyssa. So I decided to start up a company with colleagues in Indianapolis, Bob Bowen, among others, called Celsus. And this is in 2006. 2006, 2007. Before I knew it, we had, well, the CEO, not me, and the CEO is very sharp, his name was Brian Stater, had offers from 4 different investors and companies to buy the technology, which we hadn't proven yet, right? So long story short, Celsus was acquired by Moscoma, and I agreed to become their CTO for a while to transfer the technology. A while turned out to be about 5.5 years. I took a partial leave of absence from Purdue and then flew back and forth between Indianapolis and Boston every week to work on this. But what's amazing here is in January 2008, when I started, this is what it was. It's a huge team effort. This is not because of me, but just the company. By August, you had this. By November, it was running. So this is a ton a day, and this is all now reported in a DOE report on the topic, just released in last August, I believe. So all the details of it. So the point being is we scaled it up and started looking at how to do the conversion, and then I worked on that with the team for about another 4 years. I left Moscoma. I stepped down as CTO in 2013 and then left Moscoma in 2014. Came back to Purdue and became a professor, which, by the way, is the greatest job in the world, I think. So fantastic job. So the enzymes we use for hydrolysis, we really studied them, and it turns out 3 major components: cell-bio hydrolase, endoglucanase, and beta-glucosidase. 3 components. Each of the components have different types of enzymes that have different isoelectric points. Isoelectric point is where the enzyme will precipitate. You don't want that, because if you don't have all 3 enzymes, you don't get any glucose. All right? And then you have hydrophobicity. So that means they have different propensities to absorb with the substrate, with anything else that's in there. And realize when you're working with wood or corn stover, you're introducing dirt. Every once in a while, a metal, bolts, wood chips, I mean, you name it. So this is not your reagent-grade cellulose Avicel, which is ordinarily used as excipients in aspirin, but also used in labs for qualifying whether an enzyme activity is good or not. Okay, so all these properties had to be considered as we scaled up. But one thing we found very quickly is that when you pre-treated the material, the lignin had lower molecular weight components which generate a mix of phenolics. And when the phenolics were present at concentrations as little as 4 milligrams per milligram cellulase protein, and realize we're adding, our objective is to add 1 or 2 or 3 milligrams of protein, right? So that means if we have 1 or 2 or 3 milligrams of phenolics, we're done. So we had to deal with this. These are soluble phenolics. So we did some LC and mass spec and all this. And these are the phenolics that are given off by the lignin due to liquid hot water pretreatment and hydrolysis, which are designed to interfere with cellulases, because cellulases are the enzymes pathogens use to attack plants. So this is the first line of defense for plants, to prevent them from being infected by certain microorganisms, fungal microorganisms. And what's the source of our enzyme? A fungal microorganism. It's T. reesii, okay? Named after Elwyn Rees, who discovered it. So then Macmillan at NREL said, okay, let's wash the corn stover, wash away the phenols, and we should get good hydrolysis or better. And so this worked in a way. Okay, so not washed, lower conversion. You start washing out all the phenolics. Forget about the cost for now, but at least you could get good conversion. One problem. After we took the washed material, this is for a mixed hardwood, the bottom line, which is for 2.5 FPU, gave us very little conversion. The top line, which is 40 FPU, 80 milligrams per gram, you can never afford to do that, gave us very good conversion. Makes no sense. And we would run the hydrolysis for a long time, 5 days, it didn't change. The severity is the time and temperature we use for pretreatment. So higher severity means we've been rougher on the cellulose, gotten more lignin out. So if you look at this, it is very, at least to me, it seemed, why should it be that there's such a huge difference? Unless we were titrating some sort of activity into the biomass material. Now think about that. Well, it turns out, here's just some scanning electron micrographs. If you pretreat the material, you start to get formation of these balls. And if you pretreat it at higher severity, this is, I think, 15 minutes at 210 degrees C, you get even better structures. Turns out Mike Himmel at NREL identified these balls as being lignin. So here you have nicely formed high surface area lignin particles, which are hydrophobic, and you'll notice the enzymes themselves are hydrophobic. Okay. And so what we decided to do is see, okay, maybe the lignin or something in the pretreated material is absorbing the enzymes. So what we did a series of batch equilibria studies. And what we found is Langmuir relationship, no surprise. I mean, these things aren't real porous. The enzymes are really big. We also found that the affinity for protein increased with severity. The maximum capacity may decrease, but that's hard to say whether there's a trend, but inherently more absorption occurred. The partition coefficient, the propensity of the protein to go into the lignin, the absorbent lignin, also increased with severity. And then when we did gels, this is a gel where we separated the proteins on SDS gel, and we measured the different components. What we found is what really disappeared is the beta-glucosidase. So this lignin selectively absorbed the enzyme required for the last step in the sequence that formed glucose. Not a good thing, huh? And so the way this works is cellulose goes to cellobiose. So you got endoglucanase and cellobiohydrolase. The last step is beta-glucosidase to glucose. But if the beta-glucosidase is removed by the lignin, the last step will not occur. Then what happens is the cellobiose builds up, and the Ki of the CBH is 1 millimolar. Now we're trying to get 150 grams per liter sugar in our practical process, and 50% of the activity of CBH will be inhibited by 1 millimolar of cellobiose. So that's a problem. So we thought about it, and what we did is we dumped in a bunch of BSA to our cellulase prep. Anybody ever done any ELISA assay? So then what the BSA did, I'll just cut the long story short, is it coated the lignin selectively. And when it did that, the beta-glucosidase no longer absorbed. So no pretreatment, no BSA added, and look, we're running this 168 hours, and then we dilute the enzyme with BSA, and boom, we're at 90% conversion. You're not going to use BSA, obviously, but this proves the point. So we did another experiment, and remember I mentioned Avicel? So when we add it to Avicel, there should be very little difference because there's not a lot of lignin, and there isn't within the accepted pH 5.5, and what happens there, that's close to the isoelectric point, so we're precipitating out beta-glucosidase. But when we added the BSA, what you can see is the conversion appears to jump. One other way to look at it is this is a milligram cellulase per milligram protein. So if you're in biochemistry, my first project was with Dr. Vic Rodwell. I don't know if you remember who he was, but I worked on, interesting enough, on HMG-CoA reductase, which was an enzyme that forms one of the precursors for lipids, and the inhibitor for that, so statins. Okay? But anyhow, what happened is you want to have higher specific activity. Turns out for cellulase, that's not the case. Oop. So what happened is as we dilute, add more protein, the milligram cellulase per milligram protein decreases, and the conversion goes up significantly. So what we're really doing is we're diluting this protein with a non-catalytic protein. It coats the lignin, redirects the cellulase to the cellulose, prevents the beta-glucosidase from being absorbed, and gives you high yield. Now, BSA is not practical, so one of my colleagues, first slide, Professor Christian Farinas at Embrapa in Brazil, substituted BSA with soybean protein and got the same result. All right? So that then indicated there is a pretreatment conundrum, but if you then know what it is, you can deal with it. And that is, as you pretreat more severely, you expose more lignin. As you expose more lignin, it absorbs more enzyme. And although the ultimate conversion might be higher, you have to use so much enzyme it's not practical. So what you're really doing is using enzyme protein to titrate the exposed lignin, the inhibitor. So we said, heck with that. Let's just use something that's a lot cheaper, right? So lessons learned, surface area is important. I think that's chemical engineering, mechanical engineering, biomedical engineering, ag and bioengineering 101, right? You want lots of surface area, most times, right? All right? And you can read them. And so we've repeated this theme time and time again. You also have to understand the enzyme catalytic system. The next step that will really decrease the cost of some of these biorefineries is the ability to take a solid material, let's say that's at 70% moisture, and make it into a slurry. So that's what people are working on now. So you can pump it from one unit operation to the next instead of trucking it or ordering it. So the question then is, what about future research? We can read through the list, but there's all sorts of possibilities. Liquefaction, we hope that a project will soon be funded by DOE. We're under negotiation. The idea is we will take a solid material and make it pumpable using a centrifugal pump. And that was a huge issue. I can't tell you the details when I was at Moscoma. Very, very expensive capital-wise. But if you can get around that, it will decrease the cost of that first plant that must be demonstrated for this technology to be scalable. Injectable biologics, again, these are proteins at interfaces, proteins at high therapeutic proteins at very high concentrations, which might be injected. So I wish to study this, learn a little bit more about it. This is very important for cancer treatment. So instead of having an intravenous solution, the concept of the industry now is you inject it once a week, once a month, and then you have controlled release of the protein into the lymph system. Search for low-cost lignin blockers for obvious reasons. Rapid pathogen detection. That's where we started. Enzyme-assisted fractionation of corn. We've now used the cellulase technology to fractionate corn into starch, protein, germ, which is the oil, okay, and pericarp, which is the cellulose portion, with no chemicals added, using a mixture of enzymes at 50 degrees C. So that means you can now take these very large wet mills and scale them down, where places in the world that don't have other technology could use these to make some very nutritious products from corn. All right? And nanocellulose with self-healing properties. Again, this is thanks to a student that Christian sent to us from Brazil. We're now able to ferment glucose into cellulose, get a cellulose membrane or sheet, program this with some DNA. And then if we have repair molecules that have the complementary sequence, we can in fact get them, have a mixture of fluid so that different parts of the material would be programmed for different repairs with a mixture of the fluid, where the fluid would be directed by hybridization of whatever signal is at a specific part. Why is this important? It's called space shade. They're now thinking about putting up CubeSats into space. They need a big umbrella so these CubeSats can look out. And so we're thinking about this. We've actually submitted a proposal to NASA, but it got rejected. But hey, oh, I'm on tape again. Well, we'll try again, yeah. Last slide. So for those that think this is a pipe dream, it really isn't. These are the companies that are already existing in this space. This is the National Academy's report released in 2015 called The Industrialization of Biology. The products on your right, the green, are ones that are already in existence, and you can read through them. Many of them are polymers, amino acids. Some of them are for food. And the report, by the way, is online, $60 paper copy, free if you download the PDF. Just go to the National Academy of Sciences website. The ones on the left are still under development. You probably see some companies here in California. So if I were a student looking for a very exciting future, I would come to Davis, among other, I'm Purdue, of course. Of course Purdue, but since I'm at Davis, a land-grant school anyhow, land-grant mission, right, which is research, teaching, engagement. And work in this area because it's a new thing. Not everybody's going to embrace it initially until you can prove it out. That's quite healthy. But as you can see, if you really stick to the scientific technology, understand what's happening, and then do the engineering, I think we will have a very bright future. The industries here in this report, I think, were credited with economic activity around $200 to $300 billion. It could be $50 billion, but the point is it's growing very rapidly. And if you want to enter into an area, something growing rapidly. I originally got my degree in chemical engineering. I wanted to study fluidized beds. Instead, I met George Sell. I got into immobilized enzymes and then cellulases and biotech and HMG-CoA reductase. And 1976 was the first practical cloning. I graduated in 1977 and decided to switch into biotech. So here I am. It turned out okay, I think. So with that, thank you so much. It's really been a great pleasure.